Welcome to lecture on drug designing. Myself Dr. D.S. VZK Kaladar, Associate Professor, Microbiology and Bioinformatics, UTD Atal Bihari Vajpayee University, Bilaspur, Chhattisgarh. In this lecture, we will know about what is domain and motive, evaluation of Ramachandran plot, and drug designing. Coming to the terms. What is domain? Domains are distinct functional and or structural units in a protein. What is motive? A sequence motif is a nucleotide or amino acid sequence pattern that has some biological significance such as being DNA binding sites for a regulatory protein. What is quantic? A continuous sequence of DNA that has been assembled from overlapping cloned DNA fragments. What is profile? Profiles are used to model protein families and domains. What is conserved site? Conserved sequences or conserved sites are identical or similar sequences in nucleic acids, either DNA or RNA, or proteins across species, like orthologous sequences, or within a genome, like paralogous sequences, or between donor and receptor taxa, like xenologous sequences. Domains are responsible for a particular function or interaction contributing to the overall role of the protein. Domains may exist in a variety of biological contexts where similar domains can be found in proteins with different functions. An appropriate example is pyruvate kinase. If you see in this picture, a glycolytic enzyme that plays a major role in regulating the flux from fructose 1,6-biphosphate to pyruvate. It contains an overall beta-nucleotide binding domain in blue, an alpha by beta substrate binding domain in gray, and an alpha by beta regulatory domain in olive green, connected by several polypeptide linkers. Each domain in this protein occurs in diverse sets of protein families. Motif. When a sequence motif appears in the exon of a gene, it may encode the structural motif of a protein. Outside of gene exons, there exist regulatory sequence motifs and motifs within the junc such as satellite DNA. Motif representation can be done as a pattern, may be written as NPSTP, where N aspargine, P as proline, S as serine, and T as threonine. X means any amino acid except X, which is been written in flower brackets. If it is in square brackets, XY means either X or Y. Several great sites, including the first four, which are meta sites, motifs scan, interprofile, motif, CDD, CD visit, PPAM, scan prosite, prodome, smart are generally used in the motif prediction. Let us see about the differences between domain and motif. Domain. Protein domains are three-dimensional structured in or on a protein that can interact with other proteins, macromolecules, molecules, and have some biological function. If it is a motif, a protein motif is a super secondary structure consisting alpha helix and or beta sheet and random coil. It may not necessarily have the biological function. Domains are independently stable, whereas motifs are not independently stable, not stable out of the protein. Motifs can be part of domains. Domains are functional regions of the protein, whereas motifs are structural characteristics. If a motif may have biological function, it can be called as domain. Example, zinc finger motifs are related to DNA binding, so they are also called zinc finger domains. Molecular modeling. Molecular modeling is a collection of computer-based techniques for deriving, representing, and manipulating the structures and reactions of molecules and those properties that are dependent on these three-dimensional structures. Molecular modeling has become a valuable and essential tool to medicinal chemists in the drug designing process. Molecular modeling describes the generation, manipulation, and representation of three-dimensional structures of molecules and associated physicochemical properties. Coming to how the evaluation of Ramachandran plot can be done. Ramachandran plot is probably the very best indicator of the quality of the experimental determination of three-dimensional protein coordinates. Ramachandran plot is then used to assess the chemical correctness of backbone, either pi or psi angles, where the percentage of an at least 90% in the most favored regions are preferred. The pi-psi plot of the amino acid residues in a peptide is called the Ramachandran plot. It involves plotting the pi values on the x-axis and the psi value on the y-axis to predict the possible conformation of the peptide. The angle spectrum in each axis is from minus 180 degrees to plus 180 degrees. G and Ramachandran used computer models of small polypeptides to systematically vary pi and psi with the objective of finding stable conformations. For each conformation, the structure was examined for close contacts between atoms. Atoms were treated as hard spheres with dimensions corresponding to their van der Waals radii. Therefore, pi and psi angles which cause spheres to collide correspond to sterically disallowed conformations of the polypeptide backbone. In a polypeptide, the main chain N-C alpha and C alpha C bonds relatively are free to rotate. These rotations are represented by the torsion angles pi and psi respectively. Ramachandran plot generated by Procheck validation server. In the below given diagram, the white areas correspond to conformations where atoms in the polypeptide come closer than the sum of their van der Waals radii. These regions are sterically disallowed for all amino acids except glycine, which is unique in that it lacks a side chain. The red regions corresponded to conformations where there are no steric clashes, that is, these are the allowed regions, namely the alpha helical and beta sheet conformations. The yellow areas shows the allowed regions. If slightly stronger, van der Waals radii are used in the calculation. That is, the atoms are allowed to come a little closer together. This brings out an additional region which corresponds to the left-handed alpha helix. L amino acids cannot form extended regions of left-handed helix, but occasionally individual residues adopt this conformation. These residues are usually glycine but can also be aspargine or aspartate, where the side chain forms a hydrogen bond with the main chain and therefore stabilizes this otherwise unfavorable conformation. The 310 helix occurs close to the upper right of the alpha helical region and is on the edge of allowed region, indicating lower stability. These allowed regions generally involve steric hindrance between the side chain C-beta methylene group and main chain atoms. Glycine has no side chain and therefore can adopt pi and psi angles in all four quadrants of the Ramachandran plot. Hence, it frequently occurs in turn regions of proteins where any other residue would be sterically hindered. This is one of the evaluation method of Ramachandran plot using Procheck. Here the residues in the favor regions are showing 95.2%, that is, the model is valid. After validating three-dimensional structure of protein, the drug designing is the main concept where the model structure can be taken as a ligand or a receptor. Coming to the drug designing. Drug design is the inventive process of finding new medications based on the knowledge of a biological target. The two most common techniques for determining three-dimensional structure of protein targets for drugs are X-ray crystallography and NMR, that is nuclear magnetic resonance spectroscopy. There are four different methodologies commonly used in drug designing. The first one is the structure-based drug designing or direct drug designing. Second, ligand-based drug designing or indirect drug designing. Third one is rational drug designing. And fourth one is computer-aided drug designing. The first technique to be used in drug designing is structure-based drug designing. It helped in the discovery process of new drugs. Information about the structural dynamics and electronic properties about ligands are obtained from calculations. Structure-based drug designing can be divided roughly into two categories. The first one is ligand-based and second one is the receptor-based. The second one is the ligand-based drug designing. The first category is about finding ligands for a given receptor. A large number of potential ligand molecules are screened. Several ligand molecules can be available in servers like DrugBank. Quantitative structure activity relationship or QSAR is one of the most widely used approaches in ligand-based drug designing processes. It saves synthetic efforts to obtain new lead compounds. The third one is the rational drug design. Rational drug design is also sometimes referred as drug design or rational design. It is a process in which finding of new medication based on knowledge of biological target is done. It involves design of small molecules that are complementary in shape and charge to biomolecular target. The drug is most commonly an organic small molecule that activates or inhibits the function of a biomolecule such as a protein, which in turn results in a therapeutic benefit to the patient. In contrast to traditional methods of drug discovery, which rely on trial and error testing of chemical substances on culture cells or animals and matching the apparent effects to treatments, rational drug design begins with the hypothesis that modulation of a specific biological target may have therapeutic value. Types of rational drug designing methods are 3D structure of biological target or receptor-based drug designing, 2. Structures of non-active small molecules or pharmacophore-based drug designing, 3. Computer-aided drug designing or CAD, 4. Molecular graphics, 5. Pattern recognition, and 6. Receptor fit. The fourth one is computer-aided drug design. CADD represents computational methods and resources that are used to facilitate the design and discovery of new therapeutic solutions. Drug design with the help of computers may be used at any of the following stages of drug discovery. 1. Hit identification using virtual screening, a structure or ligand-based design. 2. Hit to lead optimization of affinity and selectivity based on structure-based design or QSAR, etc. Third, lead optimization, that is optimization of other pharmaceutical properties while maintaining affinity. Docking is a method which predicts the preferred orientation of one molecule to another when bound to each other to form a stable complex. Some of the docking softwares are Autodock, Dock, Gold, SwissDock, Docking Server, One-Click Docking, and iGemDock. Hence, these are the four methods it is using in drug designing methods. Coming to conclusion, researchers often combine virtual screening with other computational tools that make predictions about the activity of individual compounds, such as how they will interact with proteins. De novo drug design is an interactive process in which the three-dimensional structure of the receptor is used to design newer molecules. It involves structure determination of the lead target complexes and the design of lead modifications using molecular modeling tools. Drug design is an area of bioinformatics that focuses on drug molecules and inhibitors that can ameliorate diseases and disorders in the target manner. These are the references used for my presentation. Thank you.